Another Biology 121 online video. This is video 8, Structure and Replication of DNA. We're going to start by talking about the structure of the base and the bases in DNA and how they form together to form a single long DNA molecule. There are 5 bases in total that are involved in nucleic acids. 4 of them are involved in DNA, 4 of them are involved in RNA. So let's walk through that. 2 of them are called purines. These are on the left side of your screen, and they are double ring molecules. Adenine and guanine are the 2 purines, and you'll see that there are long, thick connection lines shown going off to the lower right on each of those from that farthest right carbon. That's where that base is going to connect up to a sugar molecule that helps it form part of the entirety of a DNA molecule. The 2 pyrimidines in DNA are thymine and cytosine, looking on the right-hand side of your screen from top to bottom. Again, there's a carbon molecule on the single ring this time that is positioned in these drawings on the lower right, and a thick black line indicating where that base, that pyrimidine base, will connect up with the rest of the DNA molecule. RNA is a slightly different molecule, and it actually uses a very slightly different base in place of thymine. The big difference is that DNA and RNA spin at different rates, which allows DNA to form a double helix, and RNA only forms a single helix. Suffice to say that in RNA molecules, RNA uses uracil as a base in replacement for thymine. We'll cover that in a little more depth in a minute. So, a single nucleotide of DNA consists of a base, a phosphate, and a sugar. So reading from right to left, we have a phosphate molecule over there, that's the P with the O's surrounding it, and then a sugar. The sugar in this case is a ribose sugar. It's a hexagonal 5-carbon sugar, and that is connected to a thymine base, or sorry, a uracil base in this case. So it's one of those single ring pyrimidines. Now, these nucleotides form together at the phosphates and sugars form up with other phosphates and other sugars in order to form what's called a sugar phosphate backbone. Now, the connection between one sugar and another happens, as you can see on the left-hand side of the screen here. We're looking at a single nucleotide with a base, a sugar, and a phosphate group, and another base, sugar, and phosphate group. And here we have a hydroxyl OH group and another hydroxyl OH group. And anytime you have 2 hydroxyls next to each other, you have the possibility of a dehydration reaction, where this water molecule is going to drop out, leaving these 2 molecules fused together with a covalent bond between this oxygen that was originally part of the top nucleotide on the left, and this phosphate group that was part of the bottom nucleotide on the left. So we'll sometimes see that depicted in these long chains. So looking on the left-hand side of the screen, we have what's called the 5' end, because we number the sugars in this, number the carbons in this sugar. It's 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. So this is called the 5' end, where the phosphate group is, of this sugar phosphate backbone. So we have a sugar and a phosphate and a sugar and a phosphate and a sugar and a phosphate and a sugar, and then there'd be a phosphate down here at the 3' end. Again, 1, 2, 3, the 3rd carbon in the sugar. And then these bases protrude out away from this sugar phosphate backbone, and these bases can interact through hydrogen bonds with these other bases that are part of another separate DNA molecule. This time, notice where here we have the oxygens kind of pointing upwards, here the oxygens are pointing downwards, because now if we count 1, 2, 3, 4, 5, the 5' end here is at the bottom, and the 3' end is at the top. So we have one strand of DNA that is running one direction and another strand of DNA that's running another direction. But, of course, they don't really lie flat like they're shown here. They form this double helix, so-called because the two molecules of DNA bind together through these hydrogen bonds, and they each spiral around each other in such a way that there's this uneven, lopsided minor groove and major groove. So on one side you have a little small space between these two sugar phosphate backbones, and the other side you have a much larger space between the sugar phosphate backbones. But we call this the double helix because a helix just means a spiral, and here we have two spirals that are interacting with each other. Moving on, we see again another depiction of this double spiral, and in this case we have different colors being used, an orange and a yellow, to represent purine and pyrimidine bases respectively. So here we have a purine base and a pyrimidine base, and what we'll find is that no matter which kind of depiction we're using, we always have a bonding formed, hydrogen bonds form typically between thymine and adenine, and between cytosine and guanine. So thymine and adenine each have 2 bonding sites, and cytosine and guanine each have 3 bonding sites, and so they form nice hydrogen bonds with each other in that configuration. Now, every now and then it's possible to mismatch those bonding pairs, but by far the most common bonding pairs are going to be that cytosine with guanine, adenine with thymine pairings. Now, in the case of DNA bonding to RNA, RNA doesn't have any thymine in it, so RNA nucleotides that bind with DNA are going to use uracil in the place of thymine. Again, notice that here we have the one side going one direction and the other side going the other direction through the backbones. Now, I wanted to share this commonly used but really bad, factually incorrect model. So some of the inaccuracies in here, they've accurately labeled guanine and cytosine and thymine and adenine, and they've accurately placed the guanines bonded with cytosines and the thymines bonded with adenines. But what's bad about this drawing is that it seems like it's symmetric, but that's not really the case. When we look at this drawing, we can see the minor groove on one side and the major groove on the other side, and then there's a major and a minor and a minor and a major, and depending on what angle you're looking at it from, these minor and major grooves are going to spiral up and down this double helix. In this particular diagram, that's not the case. They tried to make this one look symmetric. Notice also that all of those hydrogen bonding points go right down the very center of this drawing, whereas in reality, we're going to see the pyrimidine bases being much larger than the purine bases and occupying a larger amount of the space in between. So just watch out for that. We can also visualize this as a space filling model. I just want to move right past that. So when DNA reproduces itself, it does so as part of cellular reproduction. The only time DNA needs to make a copy of itself is when a cell is going to split into 2 basically identical daughter cells, each of which needs its own copy of the DNA. We call this process mitosis. You learned all about it back in 9th grade, but we're just going to do a quick review. So each of the cells, once they've split, spends time in a non-reproductive interphase before splitting again. So once the 2 cells with their own DNA split apart, they each go off and do their own thing for a while, and then each of them is going to split, making 4 cells now. So the cell cycle has this quiet or senescent period called gap 0, G0. Then there's this interphase period with gap 1, synthesis phase, and gap 2. The synthesis phase is the phase during which DNA gets replicated. And we're going to go through the reproductive process of DNA replication in the synthesis phase, subphase of the interphase in just a minute. And then after this gap 2 period passes, there's a cell division, and that's called mitosis. So this chart is really focused on the process of cell reproduction rather than on cell function. So during what part of the life cycle of a cell does the cell actually do stuff? The answer really is during the first gap phase. So in between, when the cells split apart and go dormant for a moment. And when they start reproducing more DNA to split again, this is gap 1 really is where the cell lives. That's where it's doing its thing, whatever its thing is. Heart cells pump blood, liver cells filter blood, all of that happens during gap 1 phase. So gap 1 is mostly about cell function and about cell growth. The synthesis phase is where DNA replication happens. And gap 2 is mostly about growth, is a growth phase preparing to split into new daughter cells. Sometimes also, that gap 0 thing, cells can go to sleep indefinitely. They can become non-functional but not dead. And if they are stuck in that G0 phase of the cell life, that's sometimes called a dormant cell. Sometimes cells that would become cancerous get forced into a dormant G0 phase as a defense mechanism through which your body will fight off cancer by causing those cancerous cells to stop splitting. So, quick review of mitosis. There are 4 phases: prophase, metaphase, anaphase, and telophase. They're shown in these 4 drawings at the bottom. Here, the centromeres divide up and the chromosomes clump up. The centromeres are these star-shaped figures made of protein that hold the shape of the cell. So it duplicates a second centromere, and then those 2 centromeres split up and start dividing up. So the nuclear membrane, where that protects the DNA, is dissolved. And during metaphase, the chromosomes line up down the middle. Meta is actually Greek for middle, and so that's why they call it metaphase. During anaphase, these doubled DNA chromosomes, which we'll talk about in a moment, get pulled apart by the centromeres so that half of the DNA goes one direction, half of the DNA goes the other direction. And then in telophase, the two cells form from the single cell as this split occurs and nuclear membrane is restored around each of the copies of the DNA. So again, that's prophase, metaphase, anaphase, and telophase, and here we can actually see them as microscope slides. During metaphase, all of the DNA is in one thin line down the middle. During anaphase, the DNA is being pulled apart into 2 pieces, and during telophase, we're starting to form 2 normal functional cells. I want to zoom in just on this section of this drawing, where in metaphase we can see these pieces of DNA. So in the left-hand side, this is during metaphase, while the DNA is lined up down the middle of the cell. And you'll notice that they've drawn these DNA pieces as X's. So a green X representing one chromosome, a red X representing another chromosome, a blue X, and a yellow X representing 4 different chromosomes. Now, these chromosomes are drawn as X's because they're actually double DNA. Each one of these X's contains 2 complete copies of the organism's DNA. In the next picture, we see, prophase, metaphase, telophase. No, anaphase. During anaphase, these sister chromatids get split apart, so the little middle part of the X gets broken, and what was the bottom half of this X is pulled down in this drawing, and what was the top half of this X is pulled up, causing the doubled DNA to split back into 4 normal chromosomes on the top and 4 normal chromosomes on the bottom. Of course, this represents a 4-chromosome organism going through its version of mitosis. Humans, of course, have 46 chromosomes, but nobody wants to draw all 46 of them, so we just simplify the pictures. All right, now let's talk about what actually happens during that synthesis phase as DNA gets replicated. So we have this DNA strand, the double helix over here, and it's being unzipped, causing the two different strands to split apart. One of them is called the leading strand. The leading strand is in the 5' to 3' direction. And one of them is called the lagging strand. That's in the 3' to 5' direction. So the leading strand can be continuously built, where the replication of this exposed DNA on the leading strand is going to be matched, where G's are matched with C's, and T's are matched with A's, and C's are matched with G's, and A's are matched with T's. So we're going to create a compatible matching set of base pairs to build this continuous new strand of DNA that's going to match up against this old strand of DNA. That happens continuously on the leading strand, but on the lagging strand, as the DNA unzips this way, the protein that does this build can only go from the 5' direction to the 3' direction. So as new DNA is being exposed on this lagging strand, it has to be built in little bits and pieces that are called Okazaki fragments. So this will be the first fragment that was built, while this part of the DNA in the lagging strand was still wound up inside the double helix. Then when it got exposed, a new Okazaki fragment started. And when this next piece got exposed, a new Okazaki fragment was started. And now that this piece is being exposed, there's going to be a new Okazaki fragment formed over here. Okazaki is just the name of the scientist, he was Japanese, who first discovered that that's how this works. So looking at this another way, here we have these orange blobs that represent a particular enzyme that helps do this work. And on the leading strand, we have a continuously built, looking at the bottom picture, a continuously built piece of DNA to match what's called the template strand or the original strand. And on the bottom, on the lagging strand, the template strand has to be built piece by piece. So here's the first Okazaki fragment, the second Okazaki fragment, and there's another enzyme, a second enzyme that comes in called DNA ligase and helps to fuse together these two Okazaki fragments to form a new match strand. So another view of this, what happens is that this ball opening up happens because DNA replication happens in both directions simultaneously. So what is the leading strand in one direction is actually the lagging strand in the other direction, and vice versa. So when we look at that together, what we're going to see is these bubbles of replication grow bigger and bigger until eventually they fuse together, and now we have two completely separate daughter DNA molecules. I'm going to jump back a little bit, just look at a couple more images that summarize how this works. There's a new enzyme shown in this particular picture called helicase, and helicase grabs this DNA double helix and unwinds it. So the DNA double helix is being opened up by the helicase, exposing two template strands, one the leading strand, the other the lagging strand. Typically, all of these drawings are showing the leading strand on top and the lagging strand on bottom, but of course that's just a convention of the drawing. We can also look at them as left side and right side, right? So the unwinding here is happening because helicase is coming up and splitting open these nice loose hydrogen bonds. Remember, the adenine and the thymine here, the cytosine and the guanine there, are only held together by hydrogen bonds. Those are really easy to pull apart. So the helicase just breaks in there, pops those two open, and that exposes the two template strands, which here are in sort of a bluish color, and those are matched with new DNA strands in green. On the right-hand side, that's our leading strand because it's one continuous molecule. And on the lagging strand, as new DNA is exposed, we build more Okazaki fragments to fill that out, until eventually two complete daughter molecules. So how do we go from a DNA double helix, here we can see this double helix, to the image of a chromosome that we looked at earlier, where it was shown as like a kind of blobby X? Well, what happens is that this DNA double helix, which I'm tracing around the picture, kind of anti-clockwise, it's actually wound around this protein, and this protein has its own special name. Its job is to form hydrogen bonds with different parts of the DNA, particularly with the sugar phosphate backbone. And what it does is it provides an anchor point for the DNA to wind around like a ball of string. The protein is called a histone, so it's a central histone around which the DNA winds. Looking at that here, what we can see is here's our DNA double helix, and then it's getting smaller and smaller and smaller as we kind of get farther back and look at it in context. So this blue wrapped up thing is a DNA double helix wound around a central histone, and then there are other histones, other proteins that attach themselves to the DNA and become spacer histones so that they keep these central histones spaced apart appropriately until eventually what we get is these wound up pieces of DNA wound around these protein balls, and that's what forms this chromosome of DNA. So here in this drawing you can kind of see how the chromosome of DNA is made up of all of this kind of winding bits of nucleosome. But the nucleosomes themselves are actually clusters and clumps of DNA wound around histones. So you can imagine that in this configuration, some of this DNA is kind of exposed to the nucleus, and other parts of the DNA are kind of tucked down in between histones, where it would be very difficult for your body to actually access that DNA that's way down in there, whereas there's other strands of DNA that are actually like really much more easy for your body to find and use. And lastly, we're going to go out on this picture. These are actual microscope pictures of doubled sister chromatid chromosomes. So this chromosome is actually in two parts, right? Here's one side of the chromosome, here's the other side of the chromosome, and during the anaphase of mitosis, these two will split, one to the left, one to the right. But you can sort of see how it's made up of all these little balls. And those little balls are the DNA wound around the histones in that nucleosome fashion. So I know that was kind of a long one. Please go jump on the quiz. It's not too hard. If you missed anything and need to come back and rewatch a bit of the video, I invite you to do that. I'll talk to you guys on Thursday.